Assalamu alaikum and welcome everyone to our molecular biology ASCP preparation series. This program is all about providing real life MCQs for your certification success. The MCQs we'll discuss today and in our upcoming session have been carefully collected from several years of past exams to give you the best preparation possible. I'm Dr. Loya Abdelati, certified as international technologist in molecular biology, and I'm really excited to share my knowledge and experiences with all of you through this series. I also encourage everyone taking the test to share your question with us so that everyone can benefit, all right? So let's dive in together, but don't forget to subscribe to our channel to receive all the latest lectures. Are you ready? Let's get started. The molecular science part is divided into four sections: nucleic acid chemistry, basic molecular theory, biochemical reagents, and genetics, which together make up 20 to 25% of the total molecular ASCP exam. In our previous lectures, we covered multiple choice question related to the nucleic acid chemistry. We then moved on to the basic molecular theory section, all right, and completed the replication part. Now we'll delve into the transcription section. Don't worry, for those who missed the previous lectures, I have added the links to those lectures in the bio below for your references, okay? So don't like to share and subscribe to our channel to give all the latest lecture. Let's complete. Okay, now let's focus on the question related to the transcription in prokaryotic cells and review ASCP questions from previous years. All right, let's see the first question we have today. The first question here is, how is the transcription different in prokaryotes compared to eukaryotes? The correct answer is messenger RNA in prokaryotes is not proceed after transcription. Actually, in prokaryotes, transcription and translation are coupled, and messenger RNA doesn't undergo modifications such as the capping, splicing, or polyadenylation, which are common in eukaryotes. All right, let's move on. The next question is, which enzyme is responsible for sensitizing RNA during prokaryotic transcription? All right, let's see the correct answer. The correct answer is RNA polymerase. In prokaryotes, RNA polymerase is the key enzyme that catalyzes the synthesis of RNA from a DNA template during transcription. It binds to the promoter regions and initiate RNA synthesis. All right, let's move on. The next question is, what is the role of sigma factor in prokaryotic transcription? All right, let's move to the correct answer. The correct answer is facilitate RNA polymerase binding to the promoter. Actually, the sigma factor is a subunit of RNA polymerase in prokaryotes that recognize specific promoter sequences on DNA, ensuring that transcription begins at the correct site. Okay, let's move on. The next question is, which sequence serve as the promoter in prokaryotic transcription? All right, the correct answer is -10 and -35 regions. The -10, it's called what? Primpo box, okay? And -35 regions are a conserved sequence in the prokaryotic promoter that serve as what? Serve as the recognition site for RNA polymerase to initiate transcription. Okay, let's move on to the next question. During prokaryotic transcription, what happens in the elongation phase? Let's see the correct answer. The correct answer is RNA polymerase sensitizes RNA by adding nucleotides. Actually, during elongation, RNA polymerase move along the DNA template, unwinding and sensitizing RNA by adding complementary ribonucleotides to the growing RNA strand. Okay, let's move on and see the next. Okay, the next question is what triggers termination in row independent transcription? All right, the correct answer is formation of hairpin loop followed by a poly-U sequence. Actually, in row independent termination, a GC-rich hairpin structure forms in RNA transcript, followed by stretch of uracil residues, causing the RNA polymerase to detach from the DNA. Okay, let's move on. The next question is what is the function of raw protein in raw dependent transcription termination? The correct answer is travel along the RNA transcript and dislodge RNA polymerase. Actually, the raw proteins bind to what? Bind to RNA transcript at specific sites and move toward the RNA polymerase. What happened then? Separating the RNA from the DNA template to terminate transcription process. Okay, let's move on and see the next question. All right, the next question is what is the Shine-Delgarno sequence rule in transcription? The correct answer is binds to the ribosome for translation initiation. Actually, the Shine-Delgarno sequence is not involved in transcription, but rather in translation. Okay, it is the ribosomal binding site in prokaryotic messenger RNA. Okay, let's move on. The next question is what is the primary substrate for RNA synthesis in prokaryotic transcription? The correct answer is ribonucleotides. Actually, RNA polymerase synthesizes RNA using ribonucleotides such as ATP, UTP, CTP, and GTP as the substrates, incorporating them into the growing RNA strand. Okay, let's move on. The next question is which statement is true about the initiation of transcription in prokaryotes? Okay, let's move the correct answer. All right, the correct answer is RNA polymerase binds directly to the promoter with the sigma factor. In fact, sigma factor helps RNA polymerase bind to the promoter region, allowing transcription to begin without needing a primer. Okay, let's move on. Okay, the next question, in which direction does RNA synthesis occur during transcription? Okay, let's move the correct answer. The correct answer is 5 prime to 3 prime. Okay, actually RNA polymerase sensitize RNA in the 5 prime to 3 prime direction, adding ribonucleotides to the 3 prime end of growing RNA strand. Okay, let's move on. The next question, what happens immediately after RNA polymerase binds to the promoter? It's like a gift question. Okay, let's see the correct answer. The correct answer is DNA strands are unwound to form a transcription bubble. Actually, once RNA polymerase binds to the promoter, it unwinds the DNA strand at the initiation site to export the template strand for RNA synthesis. Okay, let's move on. Which structure in RNA polymerase is responsible for synthesizing RNA? Let's see the correct answer. The correct answer is core enzyme. The core enzyme of RNA polymerase synthesizing RNA by what? By adding ribonucleotides complementary to the DNA template strand. Okay, let's move on. The next question is, which of the following is true about RNA polymerase holoenzyme? Okay, let's move the correct answer. Actually, the RNA polymerase holoenzyme includes the core enzyme responsible for RNA synthesis and the sigma factor for promoter recognition. So the correct answer here is answer B. Let's move on. All right, the question here, what is the role of NUSA in transcription elongation? Okay, let's see the correct answer. The correct answer is enhances RNA polymerase processivity. Okay, actually NUSA is the transcription factor that stabilizes the RNA polymerase elongation complex, ensuring efficient RNA synthesis. Okay, let's move on. We see the next question here. Which of the following correctly describes prokaryotic transcription and translation? All right, let's see the correct answer. Actually, in prokaryotes, transcription and translation are coupled, meaning they occur simultaneously in the cytoplasm as there is no nuclear membrane to separate the two processes. So the correct answer here is answer B. They are coupled and occur in the cytoplasm. Okay, let's move on. Here is another gift question. Which strand of DNA is used as a template during prokaryotic transcription? Let's see the correct answer. The correct answer is non-coding strand. The non-coding strand, also called the template strand, is used by RNA polymerase to synthesize RNA. Actually, the RNA sequence is complementary to this strand and identical to the coding sequence, except for uracil replacing thymine. All right? Let's move on. The question here is, what ensures the specificity of transcription start site in prokaryotes? Okay, let's see the correct answer. The correct answer is sigma factor. The sigma factor directs RNA polymerase to specific promoter region in the DNA, ensuring transcription start at the correct site. All right, so the correct answer here is sigma factor. Let's move on. The question here is, what feature of prepnow box, I mean -10 region, is essential for transcription? Let's see the correct answer. All right, the correct answer is high AT content. The prepnow box is rich in AT pairs, making it easier to unwind the DNA strand due to the fewer hydrogen bonds compared to the GC pairs. Okay, let's move on. Okay, the question here. What stabilizes the open complex during transcription initiation in prokaryotes? Let's see the correct answer. The correct answer is sigma factor. As I said, sigma factor helps stabilize the open complex by maintaining the unwound state of the DNA at the promoter region. Okay, let's move on. Okay, the question here. What is the significance of the termination signal in row-independent transcription? Okay, let's see the correct answer. So the correct answer is destabilizes the RNA-DNA hybrid to release RNA. Actually, the hairpin structure followed by a poly sequences weakness the RNA-DNA hybrid, causing RNA polymerase to release the transcript. Okay, let's see the next question. Okay, the question here. How does RNA polymerase overcome supercoiling ahead of the transcription bubble? Okay, let's move on to the correct answer. The correct answer is through the activity of DNA topoisomerase. In fact, topoisomerase relaxes supercoiling created by the unwinding of DNA during transcription, allowing RNA polymerase to move smoothly. All right, let's move on. In prokaryotic transcription, which component determines the strength of a promoter? Let's see the correct answer. Actually, the strength of a promoter is determined by how closely its sequences match match the consensus sequence at -10 and -35 regions. These matches help RNA polymerase attach better, which makes gene expression more efficient. So the correct answer here is the similarity of the promoter to the consensus sequence. Okay, let's move on. Okay, the question here is, what distinguishes the elongation phase of transcription from initiation? 15 seconds. Okay, the correct answer is RNA polymerase moves away from the promoter. Okay, actually during elongation, RNA polymerase release the sigma factor then leave the promoter and synthesize RNA along the DNA template. Okay, let's move on and see the next question. The next question, okay. What prevents RNA polymerase from transcribing non-promoter regions? Which one of them? 15 seconds. Let's see the correct answer. The correct answer is, alright, the correct answer is sigma factor's specificity. The sigma factor ensures that the RNA polymerase bind only to specific promoter regions, so preventing transcription from initiating randomly on non-promoter sequences. Okay, let's move to the next question. The next question is, which of the following contributes to the efficiency of transcription termination in prokaryotes? Alright, the correct answer is sequence-specific termination signals. Let me explain it. In prokaryotes, the transcription stops through the two main mechanisms, okay? Row independent termination and row dependent termination. In row independent termination, the RNA forms a GC-rich hairpin loop followed by a stretch of uracils, causing RNA polymerase to lose stability and stops. Okay, this is the first case. In row dependent termination, a protein called row, okay, binds to the RNA, then moves along it and RNA polymerase, catch up to RNA polymerase, forcing it to stop. Okay, so both mechanisms, row independent or row dependent termination, ensure transcription in precisely at the right spot. So the correct answer here is the sequence specific termination signals. All right, let's move on. The next question, why does transcription and translation occur simultaneously in prokaryotes? Okay, let's see the answer. Lack of nuclear membrane. Actually, in prokaryotes, the absence of a nuclear membrane allows transcription and translation to occur in the cytoplasm simultaneously, okay, enabling rapid protein synthesis. Okay, let's move to next question. Okay, the question here is, which mechanism ensures proper RNA length in row dependent termination? Okay, the correct answer is raw protein binding to a rat site. Okay, the raw protein attached to the specific spot on RNA called the rat site. From there, it moves along the RNA, chasing the RNA polymerase. Once raw catches up, it helps separate RNA from DNA. This leads to stopping transcription at the right places. All right. So the correct answer here is raw protein binding to a rat site. Okay, the question here, which mutation in a row dependent termination site would likely prevent proper termination of transcription? Okay, let's see the result or the correct answer. The correct answer is mutation in the rat site. Okay, the rat site is the crucial for raw protein binding. So mutations in this region disrupt what? Raw's ability to attach and terminate transcription properly. Okay, let's move on. The next question is why the RNA polymerase processivity critical during transcription? So the correct answer to ensure continuous RNA synthesis without dissociating. Okay, let's move on. Okay, the next question. Which characteristic of -35 region in prokaryotic promoters directly influences the rate of transcription initiation? Let's see. All right, the correct answer is distance between -35 and -10 regions. The optimal spacing between the -35 and -10 promoter regions allows proper alignment of RNA polymerase binding. Okay, so variation in this distance can decrease the efficiency of transcription initiation. Okay, let's move on. The question is during elongation in prokaryotic transcription, how does RNA polymerase maintain the vitality of RNA synthesis? All right. Okay, the correct answer is relying on Watson-Crick base pairing rules. Actual RNA polymerase ensures vitality primarily through the accurate base pairing with the DNA template. Unlike DNA polymerase, RNA polymerase lacks proofreading exonuclease activity. Okay, so it's relying on Watson-Crick base pairing rules. Let's move on. Okay, the question is, what would be the consequence of a mutation in the sigma factor that prevents its release after initiation? All right, the correct answer is reduced efficiency in elongation. All right, as the sigma factor must be released after initiation for the core RNA polymerase to efficiently progress along the template during elongation. So retaining sigma causes slower RNA synthesis. Okay, so the correct answer here reduced efficiency in elongation. Let's move on. Okay, the next question is which RNA polymerase subunit is critical for catalyzing phosphodiester bond formation during transcription? See, the correct answer is beta subunits. The beta subunits of RNA polymerase is directly responsible for catalyzing the formation of phosphodiester bonds between nucleotides during RNA synthesis. Okay, let's move on. All right, all right. The last question we have today. How does the anti-termination mechanism in prokaryotes allow transcription to bypass terminator sequences? So the correct answer by recruiting elongation factors that override termination signals. Okay, the anti-termination factors enable RNA polymerase to bypass intrinsic or row dependent terminator sequence, allowing transcription to continue downstream. Okay, this is the last question we have today. That's all for today. I hope you enjoy the video. Please don't forget to comment below if you have any question. Share the video with your colleagues and friends and subscribe to our channel to receive all the latest lecture. Salam.